Thank you, Nigel, and thank you for the opportunity to speak with you today. I'm going to tell you about targeted therapies. This is a new way of treating cancer. We've seen some spectacular successes in the last few years, and it's driving a new paradigm for treating patients, which we call personalised medicine. So the first half of the talk, I'm going to give you an overview of those two aspects and help you understand what they are and why it's important. And then in the last half of the talk, I'll focus on some projects that we're doing here in Melbourne in ovarian cancer. So you can see how we're applying this new approach to patients with this disease here in our city. I just might mention that I'm going to show you a few slides. They're technical slides. These are slides that I would normally use to talk to other scientists and other doctors. But what I'm going to do is I'm going to actually skim over them. I'm going to superficially cover them. So for those of you with a scientific background, you can follow the scientific details. But what's important are not the pretty colored slides, it's the narrative, the story that I'm going to tell. Okay. So this graph here, it's probably the most important lesson, I think, here. What this shows here is the amount of money that has been spent on developing new drugs in US billions of dollars, and that's shown in blue. And you can see how that amount of money has grown almost exponentially over the last 25 years. And then, I'm just trying to find the pointer, here we are. And then in orange, what this shows are the number of new drugs that have been approved. So what this is saying is that we're spending more and more money to get fewer and fewer new drugs on the market for patients who need them, right? And this model has become unsustainable. Now this is complicated, but I'll just take you through it. What this shows here are the various stages of drug development, phase 1, 2, 3, and then 4, or registration, is when the drug's been shown to be safe and effective, and then it's being evaluated by the regulatory agencies. Phase 1 typically is to see whether this drug is safe or not. Phase 2 gives us some early clues that it works, and phase 3 are the pivotal studies where we look at very large numbers of patients and we prove statistically that it works in the intended population. Okay? Now, what we're interested in here is oncology, which is shown in green, and I've highlighted that with these arrows. And what this shows is the success rate and then the failure rate for each of these stages. So in phase 1, about 60% of the drugs are successful. Phase 2, 30%, phase 3, 40%. And because these are cumulative, what it means is that for every drug that enters into the development process, only 1 in 14 is actually approved for use in patients. So there's a very high failure and attrition rate. And one of the reasons why it costs $1 billion to develop a new drug is not the cost of that drug, it's the cost of carrying the 13 other failures that go along with it, right? So how can we make this process more efficient? I'm going to show you how we've done that over the last few years. Now what this shows here is the survival rate of children with a very aggressive form of leukemia that used to be fatal. And what this shows here has been the progressive improvement in survival. So most of the children would die within a few years, and here the vast majority of the children are surviving. This shows the improvement in survival over the last 40 years from this disease. And this can be largely attributed by the very high participation rate of children in clinical trials, that they get access to these new experimental therapies. So today, about 80% of children who are diagnosed with cancer can be cured of their disease. And 90% of children who are diagnosed with cancer are enrolled in clinical trials. But in adults, we haven't seen that. And currently, only 6% of adults on average who have diagnosed with advanced cancer actually enroll on clinical trials. So we've got a lot more work to do if we're going to start seeing significant improvement in survival in adults, to enroll more patients on trials of these new investigational agents, which hopefully will improve survival. Okay, so what's changed? What has changed is the development of what we call targeted therapies. These are therapies in which the drug has been specifically designed to interfere with an abnormality that has happened, a molecular abnormality that has happened in the patient's tumor. It's specific to that particular abnormality. So the old chemotherapy was sort of a blunderbuss shotgun approach. But these new targeted therapies are like laser beams. They target in, and if we can inhibit those critical events, we can basically hit the Achilles heel of the cancer. And we call these targeted therapies. And there's two types. There's this type, which are antibodies. These are antibodies that your body normally raises against, say, foreign bacteria. And then there are these chemical entities which we call small molecule inhibitors. And they fall into those two classes. Now, the ones that we've developed over the last few years hit this typical picture here. So we have on the surface of a cell, this blue circle represents a cell, and this represents a receptor. And this is a molecule that's on the surface of the cell, and it sends signals through the cell and into the nucleus. And there's various pathways and various molecules. These drugs have been designed against these various signaling pathways, and they block those pathways. Now, this was the first of these so-called targeted therapies. It's called Herceptin. It was developed by the company that I worked for in the United States a few years ago. And it's been the poster child of this targeted therapy and personalized medicine approach. So what was done here is that the target for Herceptin is a receptor that is known as HER2. And HER2 is what we call overexpressed. So there's more of the HER2 molecule than you'd normally expect in about 20%, 25% of women with breast cancer. And so in this case, because they had a drug which specifically affected HER2, in this trial, what they did is rather than treat every patient with breast cancer, they only treated those patients whose tumors had this excessive amount of the HER2. So this was a selective trial, right? So we only treated the patients whose tumors had the HER2 overexpression. And in this study, what you see here in red is the improvement in survival over the black line. Now, that's not very impressive, but you have to understand that this was done in women who had very late-stage aggressive disease. What's happened since then is the drug has been developed in earlier and earlier stages of the disease. And now, in early breast cancer, this has been a life-saving therapy for many thousands of women. Right? The survival difference, I didn't bring that slide, but the survival difference in early breast cancer is considerable. Okay? So often we'll develop these drugs in patients with very late-stage disease, and then we'll work forward incrementally into early-stage disease and see better and better results. Okay? But nevertheless, this was significant to get the drug approved, and this has now been used by over 500,000 women around the world who have HER2-positive breast cancer. Now, the reason why I'm showing you this is to show you these numbers, because the prevalence of HER2 is about 25% in women with breast cancer. Now, if the trial had been done in an unselected population of patients, all comers, then it would have required 11,000 patients on the study to show this survival benefit, and it would have taken over 30 years at the then rate of accrual to enroll enough patients on the study. But because of this selective strategy, where we're only targeting the women whose tumors are HER2 positive, by definition, the prevalence is now 100% because we've enriched for those particular patients. Right? Now, this difference was seen with only 1,200 patients in 52 months. Right? So a 4-fold enrichment by screening for patients who have HER2 positive tumors resulted in a 10-fold increase in efficiency in drug development. Right? This has completely revolutionized drug development in cancer around the world. No company today would dream of developing a targeted therapy without some way of identifying the patients who, based on the biology of their tumors, we believe are likely to benefit from the therapy. This has become the de rigueur. This is now the standard. But this was the first time this had been done. Now, this was important to me as a pathologist and to my colleagues, because now the pathologists who look at the tumor tissue, we have to do these tests to show that your tumor is HER2 positive or not. So we're now involved in that decision-making process. Okay? And what happened here is that the two arms of the regulatory agency, one that approves the drugs and one that approves the tests, got together, and they made sure that Herceptin and its HER2 test were approved on the same day. Right? So this leads to this, what we call co-development of a drug and its companion diagnostic test. So this has led to this notion of personalised medicine. What we mean by that is that we are targeting particular individuals whose tumours have a particular drug target that we can address with these new targeted therapies. This is a new management paradigm, and so the aim really here is to match the right patients to the right drugs, or the right drugs to the right patients. Okay? So I'm going to give you one more recent example. Now, in a disease called melanoma, which arises in the skin of the pigmented cells of the skin, it's a very aggressive and lethal disease when it spreads. Half of those contain a particular mutation in a gene, which we call BRAF, and this is the designation we give that particular mutation. Now, a drug was developed that specifically inhibits that mutation, nothing else. It's called vemurafenib. These are the responses that we saw in the first patients treated with this drug. So these are scans. Here's the brain, which normally lights up like this, and here's the heart. But this is the tumor in the liver and throughout the abdomen. You can see 2 weeks later, there's almost complete suppression of the metabolic activity of that tumor. Here's another patient with metastases throughout the skeleton. See, most of them have gone quiet. This then translated into improved survival. You can see this yellow line here compared to the standard chemotherapy that patients were receiving up until then. This changed the management of melanoma overnight. Right? Now, unfortunately, the tumors quite quickly develop resistance to these therapies, and so the survival isn't long-term, but we're certainly seeing significant improvements in the majority of patients whose tumors have these mutations. We see no benefit in the patients whose tumors do not have the mutations. Is everyone following me? Good. All right. So this is the summary slide from this first part. What this shows is in time, the time that it's taken to develop these new therapies, which we call targeted therapies. One for a chronic form of leukemia took 40 years. Right? With this new approach, BRAF, I just mentioned, took 8 years, and more recently we have one that's called ALK that only took 3 years. Right? So this new approach is significantly shortening the time that it takes to evaluate these new therapies and get them to patients who need them. Right? But it also significantly increases the probability of success. All right? And in the case of these two here, we were able to demonstrate efficacy, in other words, that the drug works. In the phase 1 study, which is normally what we would normally be doing is looking to see if the drug is toxic or not, right? But here we're able to demonstrate efficacy in this particular case. Okay? So we can demonstrate the drug works much, much earlier in the process. Now, if you look at the pharmaceutical pipeline, what you see is it's literally pregnant with these new targeted therapies. And the success rate today is about 1 in 5. So we're seeing significant progress. So I'm just going to talk to you a little bit briefly about ovarian cancer. You probably, maybe you are, maybe you're not, aware that there's a large international consortium that is sequencing the genomes, in other words, looking at every single base of the 3 billion bases that we have in our DNA, of 10,000 different tumors, so that we can catalogue in great detail all the genetic events that happen in common cancers. If we have that encyclopedia, maybe we can turn to the right page and find the right sentence and the right letter where there's a mistake that we can then attack with these targeted therapies. Okay? And Australia has contributed significantly to the ovarian cancer effort in this regard. And that's largely through Professor Botel from the Peter MacCallum and his team, who started this ovarian cancer study where we've collected 1,800 samples from patients around the country, and a lot of those samples have been used in this effort. Now, what he's shown from that work is that ovarian cancer is several different discrete disease subsets. Okay? So it's not one disease, it's several diseases that share a common location. And so what we've done is we've taken this targeted therapy, personalised medicine approach and developed a program which we called Allocate. This is the Australian Ovarian Cancer Assortment Trial. The idea is that we do tests for a number of genes, alterations which are present in ovarian cancer, and we identify which particular patient has which particular abnormalities. And we use that information to help allocate them to the right therapy or to the right clinical trial. And this study is funded by the Victorian Comprehensive Cancer Centre. So this is the design. So we take patients who have got platinum-resistant advanced ovarian cancer. We do a range of different analysis on the sample. And depending on what we find, we can, and these are the proportions, we can point them in the direction of various potential clinical trials. So this Allocate study, for this first 2 years, is a pilot study. It's to basically demonstrate that we can do this, that it's effective and it's safe and it's acceptable to patients. We get the technology up and running and validated so that we can use it. So that'll take 2 years to do that. But once that's in place, then we can attach on these specific clinical trials. I'll just tell you about 2 of those. The first of those we call Cyclone, which is cyclin E amplified ovarian cancer. So cyclin E is like HER2. It's overexpressed in a proportion of ovarian cancers, about 10%. This is the molecule here. And the therapeutic strategy is to inhibit its partner, known as CDK2, with a molecule which inhibits that particular molecule. And what we're doing in terms of screening for those patients is we're doing 2 tests. This is normal here, so these are the cancer cells. Occasional cells are brown, which means that they express cyclin E, and they have normal copy number. In the patients whose tumours have excessive amounts, you can see all the tumour cells have very large amounts with the brown stain, and these dots represent increased numbers of the genes. So we can use this as a test to identify those patients and allocate them to the particular trial of that CDK2 inhibitor. Everyone following me? Yeah. Okay. Now another trial that we're working on is with Claire Scott here from the Walter and Liza Hall Institute. And her trial is called Biomeasures, biomarkers of efficiency and efficacy and resistance of this compound, which is known as a PARP inhibitor, in high-grade serous ovarian cancer. This is one of the subtypes of ovarian cancer. This is very complicated, but I'll just go through it really quickly. You've heard about BRCA mutations. So in patients who've got the BRCA mutation, if you inhibit this other molecule called PARP, you can kill the cancer cells. But you have to have both abnormalities. You have to have both the BRCA mutation and you have to have the treatment with the PARP inhibitor, the drug, right? So for patients who've got the mutations, we treat them with the drug. There's a way of killing off the cancer cells. And that's been shown very elegantly here by the improved survival. So what we're doing is screening, and I'm sorry for the complexity here, but this panel that we've got of the genes which are commonly mutated in ovarian cancer, these are the ones which we think will confer sensitivity to the PARP inhibitors. So it's not just one single test, like it is with HER2. We have to do a panel because it's affecting pathways that have many, many different players. And so we have to screen for them, and we're using this new technology called next-generation sequencing to screen patients in order to get them on the clinical trial of the PARP inhibitors. Okay, and then my last slide. So we've talked about targeted therapies, okay? If we're going to do targeted therapies in personalised medicine, we have to be able to hit the targets, all right? And so to do that, we have to find the right target, in other words, the right disease, the right subset. We have to use the right drug. We have to identify the right subset of the disease, so we have to do molecular profiling to identify them. We have to find the right patients, and we do that through those tests, like the overexpression test that I mentioned, and then we get them on the right trial. And I can't go into a lot of detail, but I can tell you that when you get all of these right, as we did with those 2 drugs that took 3 and 8 years, you can be almost certain to guarantee some success. Thank you very much.
Now, there's got to be some questions from that presentation. Thank you so very much, Paul. We've got time for a couple of quick ones, I reckon. So, thanks, Trish.
Thanks, Paul. That was a great presentation. If you say that the tumour can adapt or mutate quickly after introduction of a new therapy or treatment, do you introduce a new drug to counteract that new mutation, and how long does it take to then develop that new drug to counteract that mutation? I mean, it's like spiralling out of control, isn't it?
Yes. So the best way to think of cancer is it's another form of life. So the Darwinian evolution applies to tumours as well, right? So they form these mutations, and when they form these mutations, you generate genetic diversity. And that diversity gives some cells an advantage over other cells, right? So if they have an advantage, they're more likely to grow. When you apply treatment, you apply an artificial selection. And so you're selecting for cells which are resistant to it, right? And then over time, they may grow out. Now, whether a tumour does that or not depends on the treatment and the type of tumour, and then these accidents, these genetic accidents that happen. So it's a bit unpredictable. In some tumours, for instance, this rarely happens. In other tumours, it happens very quickly. We saw that with melanomas with the BRAF inhibitors, right? Ovarian cancer is sort of somewhere in the middle, but it's an inherent part of the cancer process. So how do you get around that? Well, you do what John Mills and his colleagues in microbiology have done for many years, which is use combination therapy. So you attack the tumour in various different ways to remove these various escape mechanisms for it. The problem then is, how do you combine these treatments? They're often developed by different drugs. They can have toxicity that arises from the combination, and they're very expensive. So that's the next step, and we're working on it, but it's still very early days.
Great, thanks. We've got time for just one more, and I'm going to go over here, sorry, for this lady who put her hand up a while back.
Thank you very much. I'm just wondering if you could comment on the Paragon trial. Is it one of these, or...?
Okay, I'm sorry, I can't. I'm not aware of that.
Thank you very much.
Okay.
That was a quick answer. That was good. So we will go for this other one.
I think mine was just a follow-on from the previous question, and you may have answered it. I just wasn't sure if I got the answer. So will the personalised medicine and the approach you've just shown us, will that also help with the process of dormancy or not?
With
dormancy, yeah.
When the tumours become quiet and quiescent. Yes.
Yeah.
No, we're generally looking at patients who would be eligible for a phase 1 or phase 2 clinical trial. We need to be able to see patients who have got measurable disease, so that when we can treat it, we can see that the tumour's actually responded. Eventually, when we've proven these therapies, we'll move into earlier and earlier lines of treatment, what we call the adjuvant setting, and that's what happened with HER2 and breast cancer, but I think that's still some years away for ovarian cancer.